In previous lectures, we have been discussing mass transfer in homogeneous medium, means everything is the same along every direction, and there is no difference of the medium from place to place. And that's why we have assumed that the diffusion coefficient, we call the DAB, is a constant, and we can pull them out of the gradient sign when we derive fixed second law. And the DAB usually refer to the diffusion coefficient of the solute A in a regular medium like water-like medium. That's DAB would refer to. And then now, from now on, and in the next a number of lectures, we will discuss mass transfer in heterogeneous medium. Means the medium isn't the same everywhere, and for some part you may have a higher diffusion coefficient, and in some other part you have a lower diffusion coefficient. And why this is important, or like the motivation of studying mass transfer in heterogeneous medium is you think about like what happens in the body. So for example, this is your blood vessel. And aligned here are endothelial cells. And then within the blood, you have red blood cells, and you have proteins, you have other stuff. And then suppose you have this is your solute. It wanted to go into, for example, a cell in the interstitial space. And like that. And this solute need to travel through different medium. First of all, it need to go through like the blood. Travel somewhere here. And if it interacts with or it bumps into red blood cells, it will reduce its time and efficiency of transfer. Or if it just goes through the plasma, then it have its own diffusion coefficient. And then it need to go through the blood vessel. So then here, it's like a porous membrane. So the blood vessel, they have pores on it as we discussed in early lectures. Then this pore itself present a different diffusion coefficient compared to the plasma. Then when this solute enters the interstitium space, it is another environment. And then if it wants to enter into a cell, the cell membrane is also different. And once it is in the cell, it's in the cytoplasm, then a totally different environment. So you can see for the entire process, these species or molecules experience a long trajectory, and in many steps, the diffusion coefficient are different. So that's one example of why we ever think about heterogeneous medium. So this lecture is meant to give you an overview of heterogeneous medium and why it is important. And in later lectures, we will start to talk about the technical details of how to considering each component. So now, before we wrap up, let's look at diffusion across a medium. Sorry, not a medium. Diffusion across a thin membrane. What is a medium? It's a specific medium. like to use thin membrane for most of the reason is because it represents the epithelial walls. So if you sort of cut the epithelial wall, so if you use scissors and then you cut the epithelial wall and then you flatten it out, then you basically get a membrane like this, and you will have some surface areas. We have some pores. Maybe some of the pores may look like a slit rectangular. Some pores may look a little bit different. Anyway, you have pores. Then if you have a solute needed to be transported across the membrane, then you know it can only go through places that you have a hole, you have a pore. And it cannot go through those like solid space. So then when we talk about transport across the membrane, as we discussed earlier, there's a topic of like porosity, the pore size, the pore geometry. And now in the next few lectures, we will discuss what's the effective diffusion coefficient of the membrane. So we know that if you consider the space, the bulk space, so you have a bulk. The solute diffusion coefficient in the bulk, if we assume it's a generic solute, so a generic solution, and the solvent is water-like medium, then you would have DAB. That is for bulk. Now, if you treat the membrane itself as a medium, so your medium can be a gas or a liquid or a solid or a membrane. So the membrane itself, the membrane occupies a physical space and it is a medium. Then we would ask, okay, what's the effective diffusion coefficient for the membrane? That's what we are going to ask. And then we can also think about diffusion through porous medium. Then, for example, like a sponge. If you know porosity, how do you model the flux, the concentration profile of solid, etc. So that's what the topics will be covered in the next a number of lectures. So this is for the introduction.